Alright, so I'm Devin Audebert, a student here at Keene State College, and when the idea of this project was first introduced in the beginning of the semester, I immediately jumped to water quality here in Keene due to an interest in hydrology and aquaculture. With this, I was specifically interested in looking for fecal coliform in the Ashwellet River, which runs through Keene and runs through parts of campus here. So to begin, I found a container from my home which could be used to collect about 1 liter of water, and I cleaned and disinfected it to ensure a pure water sample for isolating whatever it is that I find. I used an aluminum rod which I attached to the container to limit the amount of other potential bacterium being introduced to my sample from my hands and from other sources. Once the sample was collected, I brought it into the lab to be refrigerated, after which the water was diluted on plates of tryptic soy agar and grown for 2 days in an incubator set at 28 degrees Celsius. And you can see my dilutions here. As it gets more diluted, you can see the decrease in microbial growth. The bacteria that had grown was then plated 3 different times, each looking different from one another. And you can clearly see here how there's 3 different colony morphologies and clear difference in the bacterium which was isolated. One was then selected to be subcultured to eventually have a pure culture in hand. This required quite a few subcultures to ensure that the plate I had was indeed a pure isolate. Next, I phenotypically characterized the select microbe by conducting microscopy. This was done by simply staining a liquid culture with crystal violet and observing the shape and size of the bacterium. And you can see this here in a picture that I put into my poster. And this was taken with my iPhone through a microscope lens at 100x magnification. Next, we also looked at the plates assessing colony morphology, which you can clearly see here from my first pure culture picture. The colony morphology took a raised form, circular, and it had a very clear, distinct, shiny, deep yellow color. And as a class, we also tested for antibiotic resistance and susceptibility using various familiar antibiotic tablets, which you can also see here. This was the first day when the tablets were first placed on my liquid culture, still on a TSA plate. And this is 2 days after. You can see that they're all susceptible except for the P tablet, which is penicillin. Then I extracted genomic DNA, and the DNA was entirely sequenced by the Hubbard Genome Center at UNH. I then cleaned the DNA sequence up, trimming the technical sequences, trimming sequences from the ends by using a program called Trim-O-Matic, which we downloaded and used in class, assembling the shorter sequences into longer fragments, which are known as contigs, using a command called spades, and then annotating those contigs with identified genes using a PROCA command. Using this, I then located the 16S rRNA to identify my microbe as Pantoea agglomerans. Once I found a match and identified my microbe, I found other genes of interest, which you can see in a table that I made over here with the inclusion of beta-lactamase and a gyrase subunit. And I also used reference strands, the RIT273 and the MP2 strand to compare. I found, so in conclusion, I was successfully able to isolate and identify P. agglomerans from the Ashwellet River. This was confirmed by the microscopy, looking at colony morphology, comparison to similar strains, and the presence of relevant genes such as beta-lactamase and my gyrase subunit. Thank you.